Okay, so let's start to look at this. Oh well, that's the APSIM rotation example that I just downloaded from the website. Remember, I'm going to save it to another location. Oh, well, there's the Save As button. We'll go into this. That's the correct. I'll just call it example. We'll run it, and then we'll look at what's in there. We can do that while it's running. It'll just take a few seconds anyway. It runs for about 13 years. It actually has a weather file from a location in Brazil. It's neither here nor there. We can go through some of these details a bit later. But let's look at the tree management part of it. By the way, that only took 15 seconds to run a rotation and a bit. It's planting this cultivar, Grandis from Coffs Harbour. Is the genotype. That row spacing. It's planted on the 1st of August after the simulation. The clock starts the simulation. It runs for a harvest age of 7 years, and it received 100 kilograms of nitrogen per hectare at planting. We can look at the outputs for leaf area index. It reached about 3.5. Then it was harvested, replanted, and grew almost another complete rotation. If you want to look at the biomass trend, it's like that. DBH got up to 14 centimeters diameter breast height and height of about 19, 20 meters. Okay, so we can, just as an example, we can change some, we'll change for the 4 meter row spacing, 2 meters within rows. For argument's sake, we'll just plant it on the 1st of June rather than 1st of August, and we'll go for a 10-year rotation. 50 kilograms per hectare at planting of nitrogen. Save, I always fix save. I don't think you need to before it runs, because it should always do that before running anyway. Run it again, and we'll be able to see the output. So that you can see that the rotation length is extended. Leaf area index, all of these graphs go up to that June 2006 date. Diameters are a little bit larger, and heights are a little bit taller. So hopefully that gives you the sort of idea of basically how to get going and run a simulation. Now what I think we can do is
Phil, Phil, hopefully I'm connected in there.
Yeah, fantastic.
Hello everyone, and apologies for that, my tardiness and not having loaded WebEx. Phil, just a quick question about the allometry that's turning biomass into tree diameters and stuff. Is this something that is, are the parameters for that, or well, I think that's right, when the parameters for those relationships, are they solved within APSIM, or is that something you generate externally?
You have to generate those externally and then put the parameters in. So okay. I can show you that rather quickly. So this is the structure of the actual eucalyptus model that's running there. It can be edited, and if you want to keep those edits, you better put it into the version control system, either your own or as I'm learning through the GitHub process. But it's worth just spending a few moments looking at the structure of this. Various components, plant components that you would expect to see, like leaf, branch, stem, coarse roots and fine roots. In the version I'm working on, the improvements I'm working on now, which I hope will be improvements, I've included fruit as well. Gum nuts, for example, or if it's going to be a pine model, it'll be a pine cone eventually. So they're the components. There are biomass allocation rules, etc., within those. And within leaf, for example, there's a fair bit of physiology around not just biomass allocation, but use of CO2 concentrations that affect LAI development, and particularly stresses that come in through temperature or water or nitrogen, management of light through extinction coefficients, things like that. Nitrogen concentrations, pretty important. If nitrogen is a focus of your simulation, then you want to get nitrogen contents of the plant materials about right. Otherwise, the demand for nitrogen will be more or less than you want it to be. And then these stresses come through to the way photosynthesis works. So this simply, the root functionality in here is that a certain number of grams of carbon are fixed. Carbon, or I think it's biomass actually, is fixed per megajoule of radiation received, but that's discounted for various stresses of temperature, BPD, nitrogen, water, CO2, etc. And obviously highly related to the amount of radiation received.
Just a question, but a little bit of question, Johnny. Just a question about the nitrogen efficiency or growth efficiency, I guess is what you're talking about. One of the things that we've seen with eucalypts quite a number of times is that there's very little change in nitrogen concentration in eucalypts. They have a fairly static concentration, sort of dilute, sort of nitrogen out to the limit of its growth. And so it's quite likely that the function might be very sensitive to, or the functioning of the eucalypts might be very sensitive to nitrogen, internal nitrogen concentrations. Is there another way around that? Sort of to actually have a look at, nitrogen supply, as a, like the soil nutrient concentrations or something like that as a determinant of growth?
Yes, you can build in that functionality. But at the moment, what we have in there is it's a stress taken as what's the variable it's using?
Basically, Philip, it's the minimum. It's a relationship in the concentration of N in the leaves that is, but that value there is basically a relative value between a minimum and a critical nitrogen concentration. So the minimum N concentration is in many plants is somewhat similar to like the structural nitrogen component of the leaf. And then you have other nitrogen in the leaf that's sort of either storage or metabolic nitrogen contents of the leaves. So, and that's a dynamic part of the model in response to soil nitrogen supply and growth. So,
and it's also worth mentioning that the closeness with which you have the maximum minimum concentration, or the distance apart of those two, will determine how important concentration is or not. So if they're much the same concentrations, then it won't matter in the simulations. If it's broadly apart, then it won't be related. Then it will be related to foliar nitrogen concentrations. But at the moment, this has been set up to mirror the sorts of concentrations that are seen in grandis and those sorts of genotypes in Brazil and Australia, I should say. But that's of course where we might have genotype differences that we need to bring through for globulus or nitre or any other species or genuses as well of trees. Now, just getting back to Tom's question about the allometrics, it's simply, oh, well, you won't find that much of that in leaf anyway. That's in the stem component mainly. And diameter at breast height is simply set as a function of above ground weight at the moment in this version, the current version. But what I've discovered in the last few days is that we better change this to individuals' stem weight and make, because we want to be able to deal better with thinnings and coppicing, where the number of stems per hectare can change quite dramatically, and of course the metrics change with that as well. At the moment, I don't look back over the grandest data set that we have in there, and they're just about all around 1,100 stems per hectare. So it's a component of this model that hasn't been explored very well yet, and that's sort of what I'm working on at the moment to improve. And then I've got functions for bark thickness and underbark sort of parameters as well, wood density, those sorts of things.